Loor from University of Illinois Urbana-Champaign. Dr. Loor is a leading expert in metabolomics, and he'll talk a little bit about some of the research he's done over the past years today in our last Bair seminar series of the semester. So with that, you're welcome to take it away.
Thank you very much, Chris, and thank you everybody for coming to the seminar. Today I'm going to talk a little bit about some of the most recent area of research that we're starting to work in. That one deals with heat stress, primarily with dairy cattle. I understand Montana is not a big dairy state. Neither is Illinois, but we do good work in spite of that. So I'll talk a little bit about what led us to be more interested in gut function in the context of heat stress. And heat stress is just one of those challenges associated with climate change. Okay, but as we go through the presentation, once we start getting into more details of how cells respond to really stressors, we'll see that the response at a biological level is pretty similar, whether you have a stress that comes because of too much heat and humidity, as it would happen in the summer. The stress has to do with parturition. So we'll go through some of those aspects related to mechanisms, because then I want to talk a little bit more about what's happening in the rumen in particular. I may say some things about the intestine in the context of heat stress, and how the tissues are responding to that, and how we can potentially address those issues in the animal through nutrition. At the end of the day, really, the talk is about how we're trying to address some of these issues through feeding. Okay. So we'll talk a little bit more about certain amino acids, certain pathways that I think are beneficial in the context of potentially helping those tissues handle some of these challenges associated with stressors. Okay. Towards the end of the presentation, I'll present some approaches that we're using to try to understand first what's happening at the tissue level using omics data, sequencing data, the RNA level in particular, and how we use that to try to understand potential targets that we can challenge later through nutrition. Now, again, humidity and high temperatures are a challenge to the animal, not only to dairy cattle, also beef animals, more ruminants, and of course, in certain parts of the world, this challenge is a lot more stronger than in places where, for example, the THI index doesn't get above 70. This is from one of my colleagues in Beijing published this data on, this was from I believe a commercial farm. Okay, and as you can see here, during the summer months, those animals are exposed to, regardless of the hour of the day, they're exposed to these very high temperatures and humidity increases. Okay, of course, as the time goes by, things get a little bit better, so the animal is better able to handle those challenges. It's going to be at optimal levels, but during these summer months, really, this is what those animals, this is how they respond. Okay. So they're going to eat less, they're going to milk less, the milk quality is going to be affected, primarily milk protein content. And in the context of the use of energy for different processes during heat stress, what we need to keep in mind is that because of the need to get rid of this extra heat load, one of the first things that high environmental temperatures are going to do is reduce intake. Okay, and as you can see here, intake is not only related to growth, okay, because normal intakes provide the nutrients necessary for that. So if we're thinking about beef cattle, for example, I often say that when we're trying to measure the stress response in a dairy cow, it's easy to do because what's the outcome? Milk production. You can do these with animals in the range, with growing animals, you have to wait for a long time to really see an impact. But we know after quite a bit of research, different labs across the world, that the energy used for maintenance under conditions in which the animal is suffering from heat stress is going to increase, because that's a way in which the animal can get rid of that excess heat. If there's more energy being used to for maintenance purposes, then there's going to be less energy that the animal can use for growing. Okay, so this is obviously an important aspect of what happens in the animal. But from a physiological standpoint, and most of this is going to be with from data on dairy cattle. We've done across different labs. We've used the so-called Bair feeding approach. Bair feeding meaning that we essentially have a group that's thermoneutral conditions that is fed to the level of those animals that are heat stressed to avoid the confounding factor of a reduction in feed intake. As a way to understand, for example, how is the adipose tissue responding? How is the skeletal muscle responding? And what you have here, it's a model that is still, I think, the most accurate in terms of how heat stress itself potentially changes the physiology of the animal. Really, one of the most important aspects of what happens in the animal during heat stress, unrelated necessarily to a reduction in intake, is the fact that the skeletal muscle really is the one that provides amino acids as a source of glucose in the animal. Okay, so typically what you're going to see in a heat stress animal, you're going to see changes in the concentrations of amino acids in the plasma, not so much fatty acids. Again, depending on how severe the heat stress is, you may see changes in fatty acids, but amino acids seem to become very important in the context of all these physiological transactions in the animal. And again, glucose production is just one of them. These are data from this experiment that I mentioned before from a colleague in China. When they looked at the relative changes in the percentages of amino acids in plasma between heat, the summer months and the cooler months, this is what they essentially were able to measure. And what you see here with the asterisks, these are significant changes. For example, except for lysine, most of these amino acids that you see here actually increase in concentrations during heat stress. Okay, so I'm making a point about lysine in terms of milk synthesis because lysine happens to be one of the two limiting amino acids in dairy cows. Okay, so what we can kind of speculate simply based on changes in concentrations is that one of the physiological responses of the cow is to use amino acids potentially to generate glucose, and on top of that, the lack of lysine, okay, because of the reduction in feed intake, it's another factor that potentially prevents milk protein synthesis from remaining at optimal levels. Okay, we have an interest in amino acids in the context of some of the things that I want to talk about a little bit later on, so that's why I'm emphasizing that. The other thing that I want to emphasize is these other physiological aspects related to the oxidant and antioxidant state of the animal. And these are actual data from the same experiment. And what you can see here is that these three very important enzymes that are associated with the antioxidant response, during the summer, they actually seem to be acting at a lower, having a lower activity, okay? Whereas the markers that tell you a little bit about the production of pro-oxidant molecules actually increase. Some of you might have measured malondialdehyde. Okay, this is a typical one that we have an ELISA kit that we can use to measure those. These are a reflection of peroxidation processes of fatty acids, okay? But there's other types of reactive oxygen species and nitrogenous species that can be measured to get an idea of what's the potential for an oxidative stress state to be occurring in that animal. In a nutshell, really the cow during the summer, especially high-producing cows, are going to be under this more prolonged pro-inflammatory state, which kind of fits into the oxidative stress state. At the same time, they don't have enough nutrients that can potentially help with antioxidant capacity, and this is kind of what you see. So you have this augmented oxidative or pro-oxidant state that is going to have an impact in the productivity of the animal. Now, when it comes to heat stress and the response in beef cattle, we see pretty much the same responses, and you can see them here. I'm not going to go through those, but beef animals are going to respond with some fashion in terms of reducing intake. Of course, the availability of nutrients is going to be decreased, and amino acids also are part of that equation. In some instances, we can use markers to determine the response at the level of the immune system. I'm going to talk a little bit more about this each protein response a little bit later into the talk, but this or the change in the expression at the mean or protein level of a heat shock protein is a normal response to heat stress. But in fact, a lot of different stressors also are going to activate transcription of these proteins. And the biological function of these proteins is really to try to restore homeostasis. Okay, so they have a beneficial effect. We can use them as markers, for example. Okay, we're trying to address some sort of aspect of the stress response in the animal. So again, if we don't have proper heat abatement mechanisms, including proper nutrition, then of course the animal is going to reduce gain and potentially going to be in a catabolic state for fire longer. I'm going to talk a little bit more about what happens in the rumen, okay, in the context of digestion during heat stress and also at the level of the tissue. So I may come back and talk a little bit more about these. So as I indicated before, there are many different types of stressors, okay, that dairy cows in this particular example undergo that can lead to this inflammatory state. Of course, I'm emphasizing heat stress, but we do a lot of work with cows during late pregnancy and early lactation, so you'll see a lot of issues related to potential, issues with the reproductive tract, mastitis events, which occur fairly frequently after parturition. For example, if cows, are not able to eat normally because maybe they're overcrowded, some of the younger cows don't get access to the feed. I mean, we can have a situation in which the permeability of the gut, actually the intestine, is going to be compromised. And when you have the transport of these molecules from the gut into the circulation, those are going to trigger this inflammatory response. We often measure that through, for example, cytokines. And again, these are actual data. Again, emphasizing that in summer months, potentially you have a pro-inflammatory state, not necessarily because the animal might be suffering from some pathogen, but this could be a so-called non-steroid inflammatory response because maybe there's too many fatty acids being oxidized and creating high concentrations of reactive oxygen species, as an example. But the classical response, the inflammatory response at the level of the liver, which is really one of those key organs, not only for metabolism, but also in terms of responding to these stressors, is the use of amino acids for producing these so-called acute phase proteins. Okay, these are proteins that are triggered, they're regulated at the transcriptional level for the most part. And they're markers of some sort of stressor, okay, often related to an inflammatory event. okay? And I emphasized the point about the use of amino acids for acute phase protein synthesis in the liver, because in fact, the liver is the main source of something like serum amyloid, even haptoglobin. And these are data that we published about 8 years ago, in fact, in which we were following cows around parturition when they calved in the summer and when they calved in the spring. So this was a multi-year type experiment. And what I wanted to show here is the change, especially in the summer months when those animals calve. The simple fact of calving during high periods of high temperature and humidity triggers this immune response. In addition to triggering the acute phase response, there are other changes that occur. And obviously, I'm not showing everything that we were able to find as significantly affected, but there are a number of these mechanisms that are also related to some nutrient or micronutrient. For example, selenium is probably the one that you are most familiar with in the context of antioxidant responses. Okay, I'm going to come back to selenium in the context of a very important enzyme, glutathione peroxidase, probably one of the most important antioxidant enzymes in the body, not only livestock but other mammalian species. So we've known for a number of years that, again, heat stress it's associated with this pronounced immune response, not only at the level of the liver, which is what we've been working most to a great degree, but also the adipose and the mammary gland. Those types of organs also have these mechanisms that respond to heat stress, okay? And not only at the level of gene expression, but there have been some groups in the US that have used other technologies. For example, proteomics is a technology to measure protein abundance. And some of that work actually has identified some pathways, okay, that are triggered in response to heat stress, okay, just seasonal heat stress, right? So I'm going to talk a little bit more about this one in particular as we towards the end of the presentation. So again, after looking at all these physiological events that happen in animals that are stressed, heat stress in particular, one may wonder, okay, especially those not working in nutrition with cattle, is there a room for a nutrient or nutrients or nutritional management to help alleviate some of these negative effects on the animal, okay? And I'm not going to go through all these papers, but I just wanted to highlight that this is a very hot area of research, at least with cattle. I will say that most of the work looking at nutrients or nutrition in terms of heat stress has been with dairy cattle. My guess is, again, with dairy cows, you can measure the phenotype of the production right away. If you change, if you add more selenium, for example, you can measure a positive effect within a week. With beef, it's a little bit more challenging. What I've seen most often published is work with microbial-derived type products. Okay, I'm not going to show any of those data, but there seems to be some interest in again, using nutritional management to help those animals. So the short question is yes. I mean, there's definitely room for us to use nutrition, or what I like to call functional nutrients, okay, to potentially help those animals. And it could be tissue specific. So we'll show you a little bit about what we think might be useful at the level of the rumen as we keep moving through the presentation. But again, Nutrition 101, or in my case, I teach a second year sophomore animal nutrition class. That's where we talk about vitamins, minerals, what they do. Of course, all of these, again, you're familiar with some of these. Vitamin C is an antioxidant. You've heard about that. I mentioned selenium. That's also an important one. Some of these are either cofactors for enzymes, and you can go to the list and see which ones those are. Some are actually able to grab or scavenge some of these reactive oxygen species. And of course, when we're thinking about what goes into the animal, we can't really talk about, for example, glutathione peroxidase and selenium. There's potentially interactions, all different levels, which kind of makes it a little bit more complex. Okay, I won't talk much about potential interactions, but I do want to highlight that there are many micronutrients, because all of these are requiring very small quantities. For example, selenium is actually a regulated mineral. There's some levels above which we're not allowed to feed animals. The reason why I mentioned that is because, for example, we just published the updated nutrient requirements for dairy cattle, I think a year and a half ago. And we're keeping that selenium level the same. I'm questioning whether that's what we need to be doing, because we don't even know what selenium requirements are for different physiological conditions, heat stress being, or summer months being one of them. So I'll come back a little bit to that. But most of the work that we publish with dairy cows with heat stress pertains to some of those nutrients that I showed you in the previous page. So I'm going to show you a couple of examples, and this is a good one. Why? Because in this particular experiment, also out of that group in Beijing, they actually use heat chambers. You can actually control the pHI, okay, and you can put cows. In this case, this particular experiment was one in which they were essentially trying to replace a portion of the selenium selenite with this selenium methionine product. Okay, not everything, just a little bit of that selenium selenite, just to see whether that would be effective. Not necessarily in terms of production outcomes, because during heat stress, you don't necessarily want to help the animal maintain production. If you get a benefit, that's so be it. That's good, but you don't want to have a reduction in energy product of milk. So this is exactly what this experiment kind of showed. Again, performance was maintained, so there were no differences, and this is how much how much milk those two groups produced. But what was interesting from the physiological standpoint is how more effective that selenium methionine supplementation was in the context of glutathione peroxidase activity. Again, towards the end of that heat stress period, those animals had a higher activity, but they had lower concentrations of these type of lipid peroxidation products. Okay, so again, production was maintained, but the antioxidant response was enhanced. So that's something that we want to see. The other important aspect that came out of this experiment is that based on the selenium in the milk, it was obvious that bioavailability of selenium with the selenium methionine was obviously better, because there was more selenium at the level of the membrane. And selenium is important also potentially to prevent mastitis. And historically, that's how selenium was studied in dairy cows. Okay, should we supplement selenium, and how is that related to somatic cell count, the incidence of mastitis? Okay, so again, some very interesting data on some of the other information that you have here. For example, this is a liver-specific enzyme. Typically, when you have liver damage is when you see higher concentrations in the circulation. So the fact that this was lower meant that, okay, liver function was probably maintained or enhanced in those animals fed the selenium methionine. And of course, hydrogen peroxide, this is a classical reactive oxygen species that can be produced in tissues during stressful periods, and they were able to measure lower concentrations. Again, when you take all this data together, it kind of tells a story about bioavailability. Okay, that's something that we often don't talk too much about when we're dealing with certain feed additives. We work in the lab fairly often with rumen-protected methionine, but not, there's different products, but they're not the same. And bioavailability is something that sometimes I need to say something about. Okay. The other one is, speaking about rumen-protected methionine, this is the only experiment that I've been able to find in which a protected zinc methionine feed additive was fed during the summer months in, this was in Iran, I believe. So again, pretty severe heat load. And not only did it have beneficial effects in the context of production, but as you can see here, the inflammatory state was also reduced. And you can see the difference between these two lines here, the dotted line being the supplemented animals. They started supplementing on week 6 after calving, so they don't have that initial data, but they have a week after. Okay, so this was the period during which they fed this product. So again, positive effects. One of the, when you read the paper, one of the things that is a little bit curious, in my opinion, is the fact that the product that they were feeding is one in which I think it's a product out of Europe. So, typically when we have these sort of experiments in which a commercial product that's not necessarily fairly common, at least in the US, is being used, typically you want to know bioavailability, the intestine, and you want to know the protection in the rumen. So those are two important pieces of information that you need to have. They didn't report that. Maybe that's why it's not published in one of the traditional, research journals are good, but this is not just Journal of Basic Science, which, again, I'm sure they would have asked for that kind of information. But the point here is that these two nutrients, that's the other thing. You don't know if it's zinc or methionine that is having a positive effect. We work with a protected methionine product. There's no other mineral that is a protected methionine. Of course, zinc has many important functions at the level of the immune system, and also it's a potentially important one in the context of antioxidant response. But again, it's the only paper that we have in which methionine, protected methionine, has been fed during the summer. Now, historically, we always focused on the systemic response, and probably because it's easier to take a blood sample and, okay, what's in the blood? Of course, you can take a tissue sample, biopsy of the liver, and take it to the lab and do a lot of things. So that's, I think, why we haven't seen a lot of data at the level of the gut, rumen, and intestine. Up until, I think, the first paper that really highlighted the negative impact of heat stress on the intestine in dairy cows was published, I believe, in 2019. Okay, so fairly recent. It's a little bit more challenging to do. And this is Josh's collaboration to this presentation. So he needs to be acknowledged for that. But the reality is that heat stress, even though we can't see it, again, understanding that acidosis may be a prevalent process is that's kind of intuitive because the animal reduces intake. So potentially you have fermentation in the rumen, taking place for a longer period. If those animals reduce intake, passage rate is going to be inhibited. So there's a greater potential for pH to be lower than normal for a long time. But other aspects of epithelial integrity, either in the rumen or the intestine, you can't measure those. But again, potentially they're also part of these, overall change that happens in the animal that is heat stress. And I actually like these 4 so-called pillars. This is from the non-ruminant nutritionists. Okay, some friends in that work on those lesser species also. But again, I think these 4 pillars really capture the biology of what happens or what shouldn't happen, okay, in an animal that's undergoing, some seasonal heat stress, okay. Before I get into that with more details, let's talk a little bit about what happens during heat stress. In the rumen, I'm not going to get into the intestine. There's disruption of the epithelial lining of the intestine. That happens. but if in the rumen, this was the first experiment. This was published in 2022, last year. This was the first experiment that showed that in this pair-fed, using this pair-fed approach, okay, the ruminal epithelium integrity was just as damaged as what had been reported for the intestinal epithelium. Okay, and you can compare these heat stress animals relative to these pair-fed animals at thermoneutral conditions. You can clearly see integrity is it's pretty bad during heat stress. In this experiment, they also did some gene expression work. I want to get to those data towards the end of the presentation because they were able to identify some very unique biological processes taking place in these ruminal epithelium during heat stress. How do they explain these? Okay, well, you can read some of the thoughts that went into explaining, what could cause. Okay, they're eating less, so there should be less fermentation, right? There should be less BFAs being produced, but no, that's not what they were able to measure. In fact, there seem to be some behavioral changes, at least going back to one of the classical reviews in the context of heat stress in dairy cows. Some other data indicating, and even though during heat stress, even though those animals reduce total feed intake, they eat more often. Okay, so they eat less, they eat more often. So there's some behavioral changes, and of course, a more constant, having feed more constant for a longer period of time in the rumen, it's obviously going to keep the fermentation going. That's how they could explain this difference here. Even though, again, the intake was decreased, they were able to measure more lipopolysaccharide in the rumen of those animals. Again, most of you know that this is a very important immune activator, okay? And if you have these epithelial damage, there's, of course, these not only LDS, but potentially other bioactive molecules from the rumen can easily get into the circulation and trigger some of these systemic responses, okay? So that's what happens in the rumen of the animal, the dairy cow, but at least during heat stress. Now let's come back to the 4 pillars, okay? And I think this diagram here really summarizes very concisely the role of what microbes in rumen or intestine, and what the host needs to do, or what has to happen in the host to be able to have optimal gut function. Okay, and I just listed a few things that that we need to pay attention when we're thinking about these 4 pillars. Okay, so these are the bigger, the bigger pillars, and these are the things that we can measure within each of those to try to evaluate gut health. Okay, I'm going to emphasize this aspect of the oxidative status, or the oxidant state in the epithelium, rumen, and also the small intestine. But again, I won't say too much about the microbiota, but of course they do play a role. I didn't show you any data on the microbiota from that experiment with heat stress, but there were some changes that were unique to the heat stress. Okay, that could explain some of the increases in BFA and so on. So there are bacteria that are unique to the heat stress condition. Not many of them. But coming back to these 4 pillars, this idea of the 4 pillars, and what you see here, it's again, these are the 4 pillars, and you can see different things or indices or indicators that we can measure, okay, for each of those. This is from the non-ruminant literature, but I think if I had to start, studying this in more depth, I would use these as the baseline. Okay, in terms of what I need to be looking for, and in fact, some of these things we're looking for. And this paper was specific about amino acids in the diet. Okay, I don't work with non-ruminant species at all, but I know for a fact that just as in cattle, lysine and methionine are the top 2 limiting amino acids. Tryptophan seems to be an essential amino acid for pigs. I also know that something like arginine is potentially helpful. It's not an essential amino acid. It's a semi-essential or conditionally essential amino acid. And if you go across these are references to papers that actually reported a benefit. And the benefit could be of 3 different types. Okay, so I'm not gonna, you have the reference here, and I'm happy to provide a PDF presentation if you guys are more interested on that. But I want to highlight a couple of things. One, that glutamine and glutamine seem to be pretty effective across all these different, categories, except the microbiota. But why? Because we've known for a long time that glutamine and glutamine, they're interchangeable in terms of their metabolism. They feed into the TCA cycle. So they actually help generate ATP through the TCA cycle activity. So a lot of the things that you see, for example, at the level of the epithelial barrier, cells need to use energy to replicate. Okay, so that's not surprising that these 2 types of amino acids are very beneficial across all these different categories. Methionine, which is the one that we've worked quite a bit in dairy cows, seem to be important in the context of the epithelial barrier, but also in terms of supporting the antioxidant status of the intestine. That's important because that's how we're coming into these amino acid, rumen, and intestinal epithelium, area of research. Now, besides amino acids, and this, working with non-ruminants in terms of nutrition is a lot easier, obviously. I mean, we don't have a rumen, so we have to deal with the rumen. So there's a lot of different types of potential functional nutrients that can help alleviate aspects related to intestinal function and also the antioxidant state. Betaine happens to be one of them. The reason why I mentioned betaine is because we're going to talk a little bit about some of those nutrients that feed into the so-called one-carbon metabolism pathway, which is really 3 different pathways. So we in cattle nutrition, we don't feed betaine to any large extent. At least not in dairy yet. I can tell you that for sure. Maybe in the animals, there may be space for feeding betaine. Whatever we produce sugar beets, I don't know what that would be, but anyways. So this one-carbon metabolism pathway is one that we started exploring about 12, 13 years ago, because our idea was that methionine being the first limiting amino acids for dairy cows, again, it's not only useful for protein synthesis, but we recognize that metabolism of methionine is actually an important aspect of what we refer to as the one-carbon metabolism pathway. Now, it's singular in spite of the fact that this pathway is composed of the methionine cycle, the folic acid cycle, and the transfiguration pathway. So we started working with methionine to try to see if we could increase the concentrations of these 2 very potent antioxidants. Okay, so think about that. Methionine, an essential amino acid, is actually potentially linked with production of glutathione, which we know is the most potent cellular antioxidant in mammalian species. Okay, so that's how we got interested in these amino acid antioxidant sort of linkage. And if you remember the previous slide, they've been able to associate methionine also with support of the antioxidant response in the non-ruminant intestine. There are other nutrients that can feed into that. For example, if we were feeding betaine, I mean, that would also regenerate methionine, okay, on this methylated intermediate. Choline is something that there are commercial products of protected choline that we can feed. There's some interest in feeding protected choline to dairy cows. I'm not sure if in beef animals it's the same thing, but choline can be oxidized to betaine. So if we were looking at the liver, that's where these 3 pathways would be most active. Okay, meaning that enzymes like methionine adenosine transferase, which generates these S-hydroxymethionine, or BHNT, which is required for betaine to remethylate homocysteine, are going to be the most active. One of the things that I want to highlight that's become more evident, or that's shifted our attention a little bit from just thinking about these two antioxidants, is the fact that the process of DNA synthesis or DNA replication or cell growth also relies on methylated intermediates like serum. Okay, so these nutrients that I have here through these different pathways, again, not only seem to be important in terms of generating something like glutathione, but potentially helping cells, regenerate if there's some sort of stressor that's affecting them. We didn't always think along those lines, but I want to show you some data that seems to highlight a very unique role for those nutrients. Again, we worked quite a bit with rumen-protected methionine. This is from a review paper that we published a couple of years ago. This is a summary of the beneficial, or really the responses that we've been able to detect. When it comes to the antioxidant system, we and other groups have established that, again, when we increase the supply of methionine, especially in the last 30 or so days before parturition, we're able to increase the concentrations of glutathione at the level of the liver. So we know that it's being metabolized and it's being used to generate these potent antioxidants. And when we've measured some of the other potentially immune responsive proteins in the plasma, we also see beneficial effects of methionine in the context of alleviating the inflammatory state of the cow. Typically, the first 30 days are pretty, again, there are a lot of physiological changes. There's a normal inflammatory state right after parturition. The key is to make sure that we reduce the length during which those cows remain in this inflammatory state, because after a certain period, that inflammatory state it's no longer useful for the animal. Okay, and we've been able to see that these markers are going in the direction that tells us when those animals fed this protected methionine are in better shape, and again, they produce more milk. They're more efficient in converting nutrients into milk. We're not the only group that has been interested in using amino acids potentially to address tissue-specific type responses around parturition. Again, glutamine and glutamate, and again, a lot of this work that was published a few years back was with the notion that if we increase the supply of glutamine and glutamate, that one of the most important effects would be to increase cellular energy and to this metabolism in the TCA cycle. And these are some papers that have been published. I do have to say that when we look at the data across these different types of experiments, two of these involve rumen-protected amino acids. This one was done, I think, in intravenous infusions of glutamine. In terms of production responses, there really hasn't been a consistent benefit of some of these amino acids. And again, we can discuss why that hasn't happened. So unlike what we've seen with protected methionine and other groups have reported, other amino acids have not really resulted in this sort of consistent positive benefits. There's been development of other amino acid analogs, and in this case, this carbamoyl glutamate is called an analog because it can actually feed into the urea cycle and generate arginine. Okay, and arginine can then be metabolized to some other molecules that also help in the antioxidant response. This kind of analog has been actually pretty consistent in terms of the positive effect. So it's been an interesting set of data. More recently, there's been some interest in branched-chain amino acids. Some of you might know that the branched-chain amino acids are important regulators of protein synthesis through some signaling mechanisms. Okay, this is the only paper that I've seen. This is from Cornell, so I mean, Chris would know, Overton is here. So yeah, they published these, a couple of papers, but again, not a lot of production responses, some, benefits in certain physiological indices, but really not consistent response in terms of the the immune and the inflammatory state of the animal. Okay. All right. So let's move into a couple of slides just to show you guys how we've used some published data from RNA sequencing studies. Again, this is how we've started. Again, trying to understand a little bit using published data from experiments on heat stress involving either the intestinal epithelium or the rumen, trying to see what kind of connections, networks we can find, and whether some of those could be potentially targeted through nutrients. My former PhD student, Valentino, who's now in Italy, did this work. Couple of papers. Again, this was the first one published in 2019 that actually showed, this leaky gut event in dairy cows. Now, how do we do this, or how does Valentino do this? I don't know. Of course, we download the data, and this is sort of the pipeline that we have. Valentino, when he came back to Italy, developed this tool that he published in Bioinformatics. And again, it's an R package that allows you to take gene lists. And again, anybody can, it's a freely accessible tool. For those of you interested, you can feed your own gene list and try to generate, using the KEGG database, okay, pathway and gene networks and connections among them. Okay, so we've worked with him on that. We can do other analyses. We can look, for example, at transcription factor enrichment based on the gene expression that you're feeding. Okay, this is a tool that's, I think, freely available. The string web tool is also useful in the context of making protein-protein interactions. Some of you might have used that. I'm not going to show any of these data, but this is how we're trying to mine all these sequencing data sets to to try to understand. For example, in this example that I put together for this presentation, we took data from this intestinal epithelium heat stress challenge, and then we try to see relative to the rumen epithelium heat stress challenge data set, how many genes were in common and what was the direction of those genes, okay? Whether, in both the intestine and the rumen, they went in the same direction during the heat challenge, or whether, this is the same direction, or whether they went the opposite direction. And, once we determine, the genes that we have, for example, in the case of opposite responses, most of those, 80-some, the 127, actually behave differently in the rumen than in the intestine. Okay, and then we can do this pathway enrichment. Okay, now this is just an example of the linkage of genes that were associated with these two type of biological processes. This is in the rumen, and again, I made a comment early on that I was personally surprised to see such a high degree of activation or upregulation of genes encoding important aspects of cell cycle and DNA replication. Probably not surprising because, looking at that histological image, you can see quite a bit of disruption of those structures. So it's probably not surprising, but I wouldn't have guessed, to be honest with you, until I read this paper. And of course, something like DNA replication is not only associated with some important enzymes, but also requires some of these nutrients, some of which need to generate methyl groups, because that's important in the context of DNA replication. So, now these are some interesting data in the context of what we might be able to target from a nutritional standpoint in the context of helping those animals sort of navigate these periods of heat stress. Of course, always interested in metabolism, so I have to show these because in the rumen also, in the intestine, not a lot happening in the context of amino acid metabolism. So I'm not showing you that. So there seem to be different responses taking place in under heat stress in those two types of scenarios. As you can go through here, you can see a lot of pathways associated with metabolism of amino acids. I go back to this one here, which is linked to the production of glutathione. Okay, and again, glutathione needs to feed into the glutathione peroxidase enzyme, which requires selenium, to again get rid of these reactive oxygen species. But this is a cycle. Okay. Glutathione is oxidized and it has to be reduced, oxidized, reduced, but you need to keep producing glutathione. Okay, so a very interesting set of data in the rumen, and this, when we started looking at this set of data, we got interested in trying to understand better, potentially antioxidant pathways across the GI tract in the lactating. I would assume in beef animals it would be the same. Okay, so the first thing that we tried to do was to use enzyme activities for some of those enzymes associated with one-carbon metabolism. We did some metabolomics, transcriptomics, and this is the first paper that we published. I think this is from February, so I went through the proof, so it should be published already. So again, fairly straightforward type of questions. For example, methionine adenosine transferase, which again, it's important to generate this important methyl donor. We measure that. We measure cystathionine beta synthase. That's important in the translocation pathway. And glutathione peroxidase. I mean, there's more data that we measure, but these are the ones that I wanted to show pertinent to what I discussed before. And you can see here, and I'm highlighting what happened in the rumen relative to other sections of the GI tract, except for glutathione peroxidase, which had higher activity in the ileum. These two other enzymes seem to be very active in the rumen. Okay, so that kind of gave us a little bit of comfort in the context of what the other data we had seen with the ruminal epithelium during heat stress. The rumen is equipped, okay, potentially to handle some of these pro-oxidant states. And it likely has to do that because the rumens where you have all this fermentation and products of microbial metabolism sort of heating the epithelium. So there has to be something else that helps them protect, from those sort of negative events. I also wanted to highlight that the activity of we've done quite a bit of work with this enzyme in the liver, okay, and this activity, this level of activity, it's about 30% of what we measure in the rumen, in the liver routinely. So the liver by far has the highest activity of these enzymes involved in one-carbon metabolism, but that's the main organ where these processes occur. But if we think about the mass of the rumen in the context of the total body weight of the cow, it's about the same as the liver in terms of proportions or even greater. Okay, so even though the activity might be 1/3 of what we see in the liver, again just by size, this degree of activity of this enzyme would be very important, in my opinion, in terms of feeding into these antioxidant mechanisms. The last 3 slides I want to show you is preliminary data that we generated after we did all this work, with the GI tract. We're presenting these the ADSA meetings in Ottawa in June. So we wanted to challenge the system, and we used fistulated steers in the rumen to what we tried to do was to restrict them, okay, for about 3 days, I think, and biopsy the ruminal epithelium before, during that restriction, and then 15 days after. And we did a lot of measurements. Of course, what I'm showing here, just to set the stage, this increase in plasma-free fatty acids is normal when you go from a positive energy balance to a negative one. You don't have feed, so you start breaking down fat. fatty acids go up. So this is normal. In terms of some of the immune-related type proteins, we measure this pro-inflammatory cytokine, not a lot of difference. This was, I believe, a difference between these two and restriction, I think it approached significance. So again, I wasn't necessarily expecting to see a marked pro-inflammatory response. They were not challenged with a pathogen, they were just being restricted. But this trend kind of follows what I've seen before during heat stress. In terms of the ruminal epithelium, and I'm showing you just a couple of things. Again, we measure CBS activity, and you can see here, again, that restriction actually triggered an increase in activity, which was somewhat interesting from our perspective, because these animals were feed restricted. So something controlled, the activity of this enzyme and whether that's, the level of the gene expression, protein expression, we still are uncertain, maybe some sort of metabolite. So this response was interesting. In terms of the abundance of GP, glutathione peroxidase protein, which we know it's important in the context of decreasing these reactive oxygen species, we saw that it was actually responding in the same way as CBS activity. So potentially there's a link between these two. Remember, this enzyme is the rate-limiting enzyme in the transsulfuration pathway, which is necessary to generate cysteine, taurine, and glutathione. And of course, these enzymes require glutathione. So these data seem to indicate that they're sort of moving in the same direction. What about other immune type responses in the epithelium? Clearly, the feed restriction did trigger an inflammatory response, at least at the level of gene expression. Okay, you can see how genes were these two genes were regulated in that feed restriction. But at the same time, not only did we have this important antioxidant transcription regulator kind of follow the CBS activity, but one of its target genes was the one that responded to the highest extent during feed restriction. The way some of these transcription regulators work, or the way in which you can study them is, okay, let's look at some of the target genes and what happens in the context of your experiment. And in our context, it was feed restriction, and we had this marked upregulation. So I can't remember if we measured protein, but this again might be some sort of counter-regulatory mechanism to try to alleviate this localized inflammatory response. Okay, so things seem to be going in an interesting direction, and I highlight this antioxidant transcription factor because there's quite a bit of data in non-ruminants in particular, but we're starting to see more published data with what I'd like to call non-essential micronutrients or additives that are very rich in polyphenols that potentially can help alleviate some of these oxidative stress responses through their action on this transcription regulator. The way this works is once something like NFE202 is activated, it goes into the nucleus, binds to sequences, target genes, and then activates transcription. So we can go from, again, either this sort of feeding approach to individual polyphenols and potentially enhance the antioxidant capacity, I would say, of some of these organs, either the rumen or the small intestine. Of course, there's a big leap between feeding and then kind of coming back and acting specifically what you wanted, but I think there's potential to at least study that. And that's what I always, of course, I have to end with this with this sentence about information on the back of, because in this particular case, I think we have a very unique system in the rumen and the intestinal epithelium, one that we still don't understand very well, one that I think we can target with some of these functional issues. And with that, take questions. Thank you.
So most of the data you showed, I think, from what I was able to catch, was in Holstein cattle, and I'm high producers. I'm wondering if you have any insight maybe on how a Jersey cow may react, because that fattier milk might change the nutrient allocation a little bit.
Do you know of jerseys? No, I think we have jerseys anymore, do we?
Small amount.
Based on what I know, I think they're gonna, if they're heat stressed, if they're, stressed by some of these events that I mentioned, I think they would respond in a similar fashion. I think it would depend on the degree of the stressor, but I don't see why they would have. I know they're unique, I mean, relative to all of them, but at this level, my guess is that they're probably very similar.
Por exemplo, ele manda essa informação de custo.
No, and one of the things, that's my guess. No, and I think, this tolerance to the heat, it's obviously inherent in some of these species. So in something like Boston, because I would imagine, unless it's a severe type of heat stress, I don't think we would see the same response. I guess, would be based on, you don't know about the genetic stuff, the systems.
So on that, when you talk about the heat stress and you use THI, how likely, or are there some thresholds in there where you get no response and then you have to pick them up? I'm not real familiar with your scale, but, so they have 83, they have 70, like in Illinois, what would that be and what would it be here?
Illinois is not an example of place where where animals are gonna suffer for any extended period of time.
Right,
I have seen, especially with beef cattle, I've seen moderate THI and extreme THI. Moderate would be between 75, 76 to maybe 81, 82, and then anything higher. But at least in beef, I've seen studies looking at the degree of THI. And at least for some of the data that I showed in one of the early slides, I think at moderate THI, you see these responses. If you were to measure something in blood, like some of these heat shock proteins, they start early on. So it doesn't take a lot of heat and humidity to trigger these physiological response. To try to, again, let's come back to Illinois. I don't think, again, in July it might be challenging, maybe towards the end, and maybe the first 2 or 3 weeks of August. I don't think it gets that in.
what was the purpose of the feed restriction and the severity? So you think about feed restriction on how people feed beef cattle in a feedlot and go to restricted diets to get better efficiencies. How does your feed restriction match up with that?
Great question and great point. I don't work with beef cattle, so I was unfamiliar with the fact that they get feed restricted. The feed restriction was a challenge.
Oh, okay.
A stressor that we could do relatively easily. And we are doing a heat stress challenge. Talk to some of the guys. We're trying to see how hot it's going to get in the summer because we don't have negative control to this stress. So we may have to wait until September to do a heat challenge where we're going to try to do all of these other. But the feed restriction was simply to challenge the guys, the rumen.
And you just pulled 25% of the ad lib?
That was based on my colleague judgment plans.
Yes.
So.
I say that we'd see a pretty different effect here because unlike a lot of places, we have a really strong diurnal cycle. Like we're gonna have heat stress during the day, but it's gonna cool down so dramatically at night.
That's a good point.
Yeah, I think the impact would be mediated a lot by the cycling temperature here.
That first flight from Beijing, I mean, it didn't budge, whatever, I mean, those summer months, we just didn't get below
I
70.
mean, we can have in the summer a 40-degree swing between day and night.
And we don't have the humidity here.
Snow.
Yeah. I think,
Challenge.
Yeah. and Most
Days that are 100+ degrees, but at night it'll be 55.
Yeah, I tried to learn a little bit more about the unique aspect of cattle management in Montana, which we're counting, but it was sparse. So I can believe that it's different here.
Well, with that, to stay a little on time and give the grad students their time, we'll end the questions there.
Well, thank you very much.
Thank you very much.
Appreciate
thank
it.
you.
Thank you.